Thanks. My name's Julian Gough, by the way. So everything in this talk basically summarizes very briefly in 10 minutes what's in this paper. I'm going to talk about what it does, how it does it, the proof of it, and what you can use it for. So the orange box here, normally, is this not? This got moved. Sorry, I don't know how it's set up. Okay, is that working? It takes as an input completely blind, a single genome, so no knowledge of disease or phenotype, and it makes a predictive output of phenotype or disease for that person, and all of this is scored and ranked. This takes place within a reverse approach, so there's that input of the single genome, predicting the outputs, and then crucially there's an assessment phase where you see which of those predictions was correct. And so your final output is actually a mechanism. It's a hypothesis that the biological mechanism by which you made that prediction is in fact causal. So I'm going to very briefly say a little bit what's there. So the genome goes in. In the assessment stage, you can do this by a questionnaire, clinical annotations from doctors, or by taking a sample and doing an assay. We did all of 3 of these in the paper, and the output being the mechanism, really. So within that box, to determine whether the variant has much of an effect or not, we use hidden Markov models previously published under Superfamily and Fathom papers. We then see what possible phenotypes and diseases could be linked to using, again, something we previously published called DCGo. And then we compare all of the, for a given disease or phenotype, we compare all of the variants against a background cohort, for example, the 1,000 genomes, we take all of the variants and compare them against all of the people all together, all at the same time, we use linear algebra. We solve an eigenproblem for that, and it's something very similar to spectral clustering, which is used in image segmentation analysis. And then to compare different phenotypes, which have very different landscapes, we use a universal distribution function that we also previously published. Now, this is not better or worse than existing state-of-the-art methods, association-based methods. It's just very, very, very different. So some of the ways in which it's different is that it can work on small cohort sizes, even one individual. You need some way to do that assessment step that I said. It can identify rare or even completely unique variants because it's an ab initio predictor, basically. It has a really high false discovery rate, but it can identify multivariant combinations and even cooperativity between variants because it's not bound by the statistical limitations of association. Really, the output is it generates hypotheses that you then need to test in the lab. It explores actually 6,000 to 20,000 different biomedical ontology terms. We haven't done any of the obvious stuff. It doesn't do trios. It doesn't take into account gender, ethnicity, age, or any of that. But the main thing is it does provide causal and verifiable biological mechanisms. For the proof of this, we created our own cohort. We recruited about 2,500 people online that have their genotype data from a direct-to-consumer we call DTC, so either 23andMe, Ancestry DNA, or one of the others. We run the predictions and present them with a questionnaire tailored to them based on their predictions, and then when they answer, they get the results. We're going to take this down pretty soon because the study's finished, but it's still up if anyone has their DTC data and wants to try it. So the output of that is that statistically, in terms of it, so if you do 100,000 randomizations of the answers, how well were we able to predict them across the entire data set? This is the distribution, so it's highly predictive. That's still with a very high false discovery rate, but it's very predictive. And then now we look at how often we're correct. So at a given cutoff, 1.8% only of the predictions that we made were verified, but 0.8% is the decoy rate. So it's highly predictive and twice as good. So where it really comes into its own is you now say across all of the thousands of phenotypes that we predicted, you can see which phenotypes have the best agreement between what we predicted and the answers that people gave. Now, with this large numbers, you're going to get some pretty good matches by chance. So if, for example, we did another randomization, and if you say, for example, you take the top 40 phenotypes, only, less than half, so more than half, 28.5 of those will not be explainable by luck. or chance in the matching. So that means that more than half of the top 40 phenotypes are going to be true. We just don't know which ones. That's why you need the experimental validation step to determine which of the 40 mechanisms are truly causal to the disease. Don't have much time to go into this, but this is a breakdown of what contributes to the scores and how we can see multivariant and combinatorial effects. What's very common is we see one required variant, which is not sufficient alone, but then a pool of other supporting variants, which are different in different people, which combined together are predictive. So in the paper, we have a whole bunch of examples that we went through. I'm just going to rip through these. This is an example of a known variant from a GWAS study that we discovered, but we were able to discover this with no knowledge or encoding of any of the results of the GWAS study. So we completely, essentially got the same output without the GWAS study. Here's another one, which is a novel, completely novel gene associated with the disease. This is a novel variant in a gene, but it's in a family that's known to be related to the disease. This is just a novel single variant. This is a novel variant, but it's in a gene that's already known. And this is a combinatorial effect. Whilst we're able to do this with this relatively small data set, we see very few of them. But there is one example here where by far the top prediction, which was correct, involved 4 variants, which there's cooperativity in pairwise between 2 of them. This last example I've got a whole slide on. This is an experimental validation done by our collaborators at King's in London. This is the HIPSCI database, so about 800 genomes, exomes, and with cell-banked 800 IPS cell lines. So we were able to make a cellular phenotype prediction here of centrosome duplication, and our collaborators ordered the cells and did this assay to check. And they saw that of the 5 out of the 800 that we predicted with this phenotype, 3 statistically significantly had a higher than usual centrosome count. So this is a highly unusual phenotype. And so we've very clearly identified this, and there's a nice causal explanation and a mutation in a binding site that's well documented in the literature and is likely to change the specificity or affinity of this in its pathway. So what could we use it for? Well, the fundamental value proposition is essentially, this is just extremely different from other things and non-competing. So if anyone that has a data set, you've run your association, you've run all the state-of-the-art tools, this can be applied for novel discovery and see things that people haven't seen before, could lead to drug target discovery. And because of the multivariant capability, it gives us alternative ways to do stratification for precision medicine. So I'd just like to thank my collaborators. This work was in about 10 years in the making. So I want to single out, when I was at the University of Bristol, a specific mention to Jan Zauke at the MRC Laboratory for Molecular Biology in Cambridge, where I am now. I'd like to single out Chang Lu. I'd like to thank our collaborators at King's in London who did all the experimental work, and a special thank you to Bastian, who helped us recruit many of the people for that direct-to-consumer cohort study. Thank you.
So same strategy, or we go one question. Alex, maybe you can.
Hi, thanks for the talk. Just a quick question about, so the output of this model is something like an individual profile score, something that's analogous to polygenic risk score profile, is that right?
No, not really. It's, yeah, it's all in the paper. I mean, we use, those 5 things that I said, and they come together, and it basically goes through this linear algebra eigen problem. So it takes into account multi-variants at the same time across many people in that landscape, but I wouldn't call it anything like a polygenic risk.
So I was just wondering about the kind of the total variance explained in terms of R square, just to put it into perspective, how good your model is in terms of explaining the variance. Do you have a metric like that?
I mean, no, I wouldn't frame it in that context. I mean, that slide where, very poor, I would say.